Contents of the cell, organelles of making stuff and moving stuff around. What you see here at the left, before I move on, is an electron a transmission electron microscopy image of rough endoplasmic reticulum. And if you recall from the beginning of this series where I introduced the organelles, rough endoplasmic reticulum is rough because it looks rough under a microscope. Those things that make it look rough are ribosomes. So here is the membrane of the rough ER, and each one of these little black dots are ribosomes. Look how densely packed rough ER is with ribosomes. You have ribosomes all over your cell. They're free in the cytoplasm as well, but they can also be found bound to the rough ER. We'll see all this in a bit. So if you recall, ribosomes are made up of small bundles of both proteins and specially made RNA for ribosomes, rRNA. rRNA and these is made in the nucleolus. The job of the ribosome, again, as we've previously discussed when we talked about translation, the job is to take mRNA and make proteins for the cell to use. That's translation. What was transcription? Do you recall? Transcription is turning a nucleic acid language into another copy of something in a nucleic acid language. So in this case, we're talking about DNA to mRNA, messenger RNA. Translation is turning something in one molecular language into something else in a different molecular language. The nucleotide language of mRNA to the amino acid language of proteins. Proteins are the actual functional working units of the cell. When you're talking about things going and moving around in the cell and doing stuff, you're talking about proteins. All enzymes are proteins. You build things with proteins. You do things with proteins. So in a way, you can think in the whole process of the central dogma, DNA codes for proteins. Proteins is the final and desired product of DNA transcription and then translation. Some ribosomes I mentioned are free and others are bound to endoplasmic reticulum, specifically rough endoplasmic reticulum. For the purposes of this exam, you should write out, if you have to write out an answer that talks about endoplasmic reticulum, write it out the first time you mention it, so that I know that you remember what ER stands for. Then after, and for the rest of the semester, unless I specify otherwise, then afterwards, it's okay to say or write ER. Okay? So rough ER, endoplasmic reticulum, is continuous with the nuclear membrane. Remember that the nucleus is bound with a lipid bilayer. Here's your nucleus. And this red line is a lipid bilayer. See, two lines. The rough endoplasmic reticulum is immediately adjacent to the nucleus. And so this, and I'll do this in a different color just so you can see it branch off, but it really is the same membrane, the same lipid bilayer. It simply comes off like that. So it's a continuation of the same membrane. The membrane undulates a lot and is maze-like to create what's called cisterns. And cisterns are these sort of dents. The undulation of the membrane is to increase surface area. Things happen and things are bound to membranes. So the more membrane you can have packed along a given length, the more stuff you can do. Sensing a theme here, right? The ribosomes make proteins. So mRNA, you have that nucleus again, with the continuation of the rough ER. When the mRNA is made inside the nucleus and leaves through a nuclear pore, there's ribosomes sitting right there on the rough ER waiting for it. So the ribosome is going to take that mRNA, feed it through like ticker tape, bring in tRNAs that are bound to anticodons and amino acids, and then string along the primary structure of a protein. The primary structure of the protein will spontaneously fold into the secondary form. To some limited degree, it can then fold more based on interactions of available R groups. Those proteins are then going to be transported to the Golgi apparatus, otherwise known as the Golgi body, to be modified and then exported by transport vesicles. We'll talk about the Golgi in a bit. The rough ER also makes phospholipids and integral proteins to build and restore and maintain the plasma membrane. So rough ER makes proteins, phospholipids, and specifically integral proteins. For the plasma membrane, and these other proteins for whatever. Very scientific term. Smooth endoplasmic reticulum is called smooth because it looks smooth under an electron microscope. You can see the difference between this and this, right? It does not have ribosomes studding its surface. There are no bound ribosomes to smooth endoplasmic reticulum. You may find them nearby, off to the side in the cytoplasm, but they're not bound to the membrane. The smooth endoplasmic reticulum's membrane is continuous with the rough ER. So here you have your nuclear membrane. Continuous with it is rough ER. And then continuous with it is smooth ER. It's not necessarily arranged at a 90-degree angle like this, but that just is easiest to draw. This membrane is also maze-like to create cisterns, again, surface area being a thing. It has no ribosomes of its own, but it is packed with enzymes, so other protein-doing molecules, to work with large molecules that have been ingested. It works with those large molecules to modify them, to break them down, to turn them into usable things for the cell. It brings in lipids, fats, cholesterol, which is like a big sort of, cholesterol and steroids are also sort of fatty acid based. Glycogen, which is that big, huge glucose chain, starchy type thing, and drugs. This is a really important detoxification organelle. In your liver, which I hope you know is a detoxifying organ, when you look at those liver cells, hepatocytes, they are rich in smooth ER. Another really important function of smooth ER is that it stores calcium ions and releases calcium ions for the purposes of muscle contraction. When we talk about muscle contraction at a molecular level later on, and we will in obnoxious detail, we're going to talk about the movement of calcium ions. When you think, where do those calcium ions come from? On an immediate basis, they come from within that same muscle cell's smooth endoplasmic reticulum. As I mentioned, the Golgi apparatus takes proteins from the rough ER and does stuff with them. This is often referred to as the UPS center or like a FedEx store, a packing and shipping center for the cell. It's on the other side of rough ER from the nucleus. So you can see the nucleus is here. The rough ER is right up against it and continuous with it. I should change this away from blue so you can see it better. It's not much better, but okay. You have smooth ER over here, which is continuous with the rough ER, but not necessarily up against the nucleus. The Golgi is its own separate thing. We're not talking about continuous membranes anymore. It's its own separate thing. Now, that membrane is made of the same stuff as the ER. It's just not connected. And it's going to be adjacent to, next to, the rough ER. The side of the rough endoplasmic reticulum that faces the rough ER is the cis face, and each of these sacs of the Golgi, I gotta change that color again. Each of these sacs of the Golgi, the side that faces the rough ER is the cis face. Cis is a chemistry term for same-sidedness. Chemistry or physics or, I don't know, both. It means same-sidedness. Culturally, we hear this a lot in cisgendered, like we are gender, we identify with the gender that our sex is. It's all on the same side. It's all sort of aligned. The trans face is the side that faces away from the rough endoplasmic reticulum. Again, we don't care so much about smooth ER right now. We're just talking about its relationship with the rough ER. Trans means away from or opposite from. So if culturally we hear the term transgender, that refers to feeling gendered opposite from what your genetic or chromosomal sex is. So this Golgi apparatus is a membranous network of flattened sacs. Again, it's often likened to like a UPS warehouse or something. It receives transport vesicles from the rough endoplasmic reticulum along its cis face. So your rough ER made all these proteins. Another thing that it does with those proteins is it bubbles them up in a vesicle so that that protein can move somewhere else in the cell. It's not just going to travel loose anywhere. Once in the Golgi apparatus, it'll move through this series of flattened sacs. Each time it moves through one of those flattened sacs, it gets modified somewhat. So you can see here you have this little three-pronged modification that it came with. And then it turns into a one-prong, and then the Golgi modifies it into a two-prong, and then it modifies it into a slightly different-looking three-prong guy. They should have given more variation for the illustration, but the point should be, the take-home point should be that it enters one way, and it leaves another way. The protein's tertiary structure and quaternary structure are largely created here in the Golgi apparatus. It modifies their shape, and it also sometimes adds proteins to it. So it assembles them or adds some sort of markers. Finally, it will tag that protein for export. Proteins are exported by vesicles along the trans face. Notice that the trans face is facing and nearby to the plasma membrane of the cell. From there, there's only a relatively short distance for this new vesicle with the final protein to be exported. If that protein's destiny is to do something inside of the cell and required Golgi modification, it could just stay. That's fine too. Together, there's a combined term for all of these membranes. The nuclear membrane, not the contents of the nucleus, but the nuclear membrane, plus both endoplasmic reticulum, smooth and rough, plus the Golgi, combined are referred to as the endomembrane system. Finally, a quick once over about peroxisomes and lysosomes. These are vesicle-bound organelles that you find. They're really just sort of bags of other contents. that you find that are going to be moving around the cell doing work on an as-needed basis. Peroxisomes help to disarm reactive oxygen species, otherwise known as ROS, especially hydrogen peroxide. Reactive oxygen species and including hydrogen peroxide, are major, they wreak a lot of havoc within the cell. They're byproducts of breaking down other things and byproducts of metabolism on sort of a more gross scale. And they're lacking a lot of stuff. They're not terrifically stable in and of themselves. So they're really inclined to bind and make interactions with whatever is nearby. Whether it harms that thing or not, the ROS doesn't care. It just needs to bind. So you don't want to have terribly many of those floating around. Peroxisomes help disarm those, basically make them neutral and make them inert. Peroxisomes also help to break down long-chain and branched fatty acids, as well as some proteins. These can then be fed into the smooth endoplasmic reticulum for further breakdown, or they could be fed to lysosomes to just get rid of it. Peroxisomes are abundant everywhere in the body. You need a lot of these to keep your cells and your cellular components safe, but they're particularly abundant in detoxifying organs because of this effect primarily and this effect secondarily. Lysosomes are vesicles that contain digestive enzymes and have an acidic internal environment to break down lots of stuff. So the pH inside lysosomes is lower than cytosol. This is your main digestive organ of the cell, the garbage heap. Whatever your cell doesn't need anymore gets thrown into the lysosome, and the lysosome attacks it with acidity and attacks it with enzymes that break it down into smaller parts. It can then release that waste into the extracellular environment, or it can recycle some of those components to use for other things inside of the cell. Whatever the cell ends up needing is what the lysosome will do with it. They're abundant everywhere. They're particularly abundant in many immune cells. Ingested bacteria and viruses that your cell's trying to attack and get rid of are going to be attacked and absorbed by lysosomes. These play a key role in demolition, breaking down the cell itself and incoming invaders, as well as autolysis, which is the deconstruction of the cell in a methodical way while it dies.